Hi viewers, now we discuss about the structure of DNA. DNA is deoxyribo nucleic acid. Now the objective of this present lecture is to understand the historical background and importance of DNA, to know the structure and components of DNA, to differentiate aDNA, bDNA and zDNA. And regarding the historical perspective of DNA structure, Linus Pauling, first he described DNA having the triple helix mode. According to Linus Pauling, DNA has three strands. So he described it as the triple helix model. And later, Erwin Chargaff, he proposed Chargaff rule. According to this rule, the amount of purines in DNA is equal to the total amount of pyrimidines. That means adenines and guanines both are purines, and thymine and cytosine are pyrimidines. That means A plus G is equal to T plus C. That means the amount of A is equal to T, the amount of C equal to G. Total number of adenines equal to total number of thymines, total number of cytosines are equal to total number of guanine. So it is called Erwin Chargaff rule. And later, in a breakthrough study, Rosalind Franklin and Wilkins, they used X-rays to reveal the structure of DNA. With their X-ray diffraction studies, they focused X-rays on DNA and they obtained the DNA X-ray crystallography structure. Based on this X-ray crystallography structure, J.D. Watson and Crick proposed the double helical model of DNA. Here we see the double helical model of the DNA, which was first described by Watson and Crick. For that, Watson and Crick got the Nobel Prize. And what is the importance of DNA? DNA or deoxyribo nucleic acid is a polymer, and it is found in the chromosomes in the nucleus and also in the mitochondria and chloroplast like organelles. DNA carries the genetic information. DNA is called as genetic blueprint of the organism because it has the total information of the structure of the organism. So it is called as the genetic material. And also DNA is transferred to one generation to another generation. It carries the genetic information. So it is called as the genetic material. Chromosomes are called as the carriers of DNA as they carry the DNA. DNA is packed with the help of the histone proteins in the supercoiled form in the chromosomes. So here we observe the two strands of the DNA. They are helically coiled, and later they are coiled around the histone proteins to form the nucleosomes, and later these nucleosomes also they are coiled to form a supercoiled structure, and finally chromomeres, and they are packed finally in the form of the chromosome. So now we look into the double helical model of DNA. As per the proposal of Watson and Crick, there are two strands of DNA. Both are arranged in anti-parallel manner. Here, one end of the DNA is called as the 5' end, and another end of that strand is called as 3' end. and in the opposite strand it is arranged in the opposite manner, like this tip is called as 3' end and another tip is called as a 5' end. So here these two strands are arranged in the anti-parallel manner and they are helically coiled. Here they are attached together with the help of the nitrogen bases. Here we see adenine and thymine. Both are attached together with the help of the hydrogen bonds. Here nitrogen bases they form the pairs. Here we observe the pairs of nitrogen bases. They are paired with the help of the hydrogen bonds. So this is one pair of nitrogen bases and this is another pair of nitrogen bases. They keep the two strands together. And here we see the two strands of helically coiled DNA, and here we also see the deoxyribo sugars. Here we see the ball and stick model of DNA. This is major groove and this is the minor groove. And DNA composition. DNA is a nucleic acid polymer and the monomers of the DNA are called as nucleotides. In each nucleotide, we can observe nitrogen bases like purines and pyrimidines, and also there is a sugar compound that is called deoxyribo sugar. Here, ribose is a pentose sugar. It is a 5-carbon sugar, so it is called as pentose sugar, and since it lacks one oxygen, it is called as deoxyribo sugar. And nitrogen bases and sugar are called as nucleosides. In combination with phosphates, nucleoside and phosphate together called as nucleotide. Now we see the nucleoside, a single nucleoside. Here this is a nitrogen base and it is bound with the ribose sugar with the help of the glycosidic bond. So this is the ribose sugar because in the second carbon position and in the third carbon position we observe two hydroxyl molecules. And now we see the deoxyribo sugars. Here these are also called as deoxyadenosine 5-phosphate because here adenine is bound by the deoxyribo sugar with the help of the glycosidic bond, and finally these two bind to the phosphate. So this is called as phosphodiester bond. Now we see another deoxynucleotide, that is guanosine. It bound with the deoxyribo sugar with the help of the glycosidic bond, and finally they bound with the phosphate. Here we see the cytosine and here we see the thymine. In all these nucleotides or deoxynucleotides, we observe the change of the nitrogen base. In all the deoxynucleotides, phosphate and deoxyribose sugar is common. The only one which changes is the nitrogen base. And here we see adenine. It forms the adenosine nucleoside and 2-deoxyadenosine, and guanine, it forms guanosine nucleoside and 2-deoxyguanosine, and cytosine, it forms nucleoside cytidine and deoxynucleoside called deoxycytidine, and uracil forms uridine, and in DNA we don't find the uracil, and thymine, it is not found in RNA and it is found in DNA in the form of 2-deoxythymidine. So here we see the two strands of DNA. These are the nitrogen bases, thymine and adenine. They are bonded by the two hydrogen bonds, and guanine and cytosine, they have 3 hydrogen bonds, and adenine, thymine 2 hydrogen bonds, and guanine, cytosine 3 hydrogen, A, T, G, C. And here each nitrogen base forms a glycosidic bond with the deoxyribo sugar, and finally it binds to phosphate. And here two adjacent nucleosides are joined with the help of phosphodiester bonds with the phosphate. So this is one polynucleotide strand and this is another polynucleotide strand. These two strands are bonded with the help of the hydrogen bonds between the nitrogen bases. Here we see thymine binding with the adenine with the help of two hydrogen bonds. Here we see cytosine binding with the guanine with the help of three hydrogen bonds. And now we see three different types of DNA: aDNA, bDNA, and zDNA. And left to right, aDNA, bDNA, and zDNA. We see the most common form of DNA is bDNA that are observed in the organisms, but occasionally we can find aDNA. It is a dehydrated form of the bDNA. And zDNA, here we see it is in strands are observed in the zigzag manner. Now we see the graphical pictures of aDNA, bDNA, and zDNA. And the advantages of double helix model is the stability. It is protected with from the attack of the H2O, soluble compounds, and H2O itself, and it provides easy mechanism for replication. The two strands which are helically coiled can be easily separated by this model.